Hey Beeks, I hope you're doing well. I hope you were able to start genetics yesterday by doing your study assignment, and hopefully that'll pay off for you today. So if you hear anything crazy happening, my daughters are upstairs playing. It sounds like an earthquake down here. But anyway, so what you're going to do today is you're going to go through a document with me, and I'm going to solve some problems with you, some introductory genetics stuff. And then that should prepare you for the end of the document where there's 5 questions that are practice questions. Now, some of you will just be, boom, you're ready to go right away, and others of you are going to be a little bit confused still. There's always different levels. You know, you'll all get there eventually, but some kids get it right away, and some kids it takes them a little bit to really catch on what's going on. My advice to you, though, is go back over your study assignment now again. The vocabulary is huge, right? Once you understand the vocabulary, then you can wrap your head around what the concepts are. And once you can do that, then you can practice. And like I said, practicing is how you learn genetics, doing problems. So if you can print the document, that's probably best, but I know some of you might not have printers at home. So if you want to open up the document that I shared with you and put it on the side of this video or something like that, I'm going to go ahead and go through the document, and every now and then I'll pause it, and I'll be looking at the piece of paper. Okay, and that's what you could do too, is just solve the problems on a piece of paper so that way you understand what's going on. Okay, so here we go. Let's get started. Okay, so here we go, Beeks. This is Introduction to Genetics, and I should have shared this document with you. Okay, so take a look in the classroom and make sure that you have this opened. Like I said, if you could print it, that would be great. And if you can't print it, that's okay. You could just solve the problems on a piece of paper. So we start at the beginning here, and people were thinking about genetics, obviously, for a very long time. And to us, some of this seems a little silly. Like, how did they not figure it out quicker than this? Hippocrates, you could see, thought about pangenesis. Like maybe you had little parts from your body travel to your sperm and your eggs, and they get passed on to the next generation. And they didn't even necessarily know what sperm or eggs were back then. They just said, oh, these little pieces collect in your body and get passed along. Some people actually thought there were even mini humans inside your sperm cells, for example, that just grew bigger instead of developing and things like that. But the blending hypothesis is the idea that hereditary materials contributed by the male and female parents mix in forming the offspring, similar to the way that blue and yellow paints make green. So this is what the main thinking was in the early 19th century. And you might be thinking to yourself, hey, that's kind of accurate, right? This blending hypothesis. You are a blend, of course, of your parents, but it's not like you take them and you mix the traits together. It's much more complex than that. So we went along here to Gregor Mendel. And if you watched the video yesterday that I shared with you, you could see that he is pretty much known as the father of genetics because of his work with pea plants. Now, they did say that his data was a little bit too accurate, but we'll go ahead and give him a break there because he seems like an alright guy. So heredity is the transmission of traits from one generation to the next, right? This genetics basically is the study of heredity. And this Augustinian monk, Mendel, you read all about him, and he was a very good mathematician. He got lucky, right, because his parents didn't have a whole lot of money, but he was a good mathematician, he was a good scientist, and he decided to become a priest, and he taught some high school biology, so God bless him. And what he did was, in this garden, which actually, funny enough, the choral kids were on a field trip years ago that I went with them to Germany and Austria and parts of the Czech Republic and things like that. It was a really cool trip, but we actually drove right by the exit for Brno, which is where this Abbey garden was. But unfortunately, we didn't have enough time to stop. But that would have been pretty neat to see where his experiments were conducted. But anyway, he said that heritable factors basically retain their individuality generation after generation. Okay, so he said basically you're not blending, right? It's not like you're taking these two paints and mixing them together. Okay, because if you had green paint from yellow and blue, well then when you go to have a kid, you're going to be passing on green, right? Not yellow and blue. And Mendel said, no, genes are more like a deck of cards, okay? And he didn't use this example back then. But think about it. The cards don't change. Now your hand could change depending on what cards you got, okay? But the cards themselves are the same. You can sort them differently and you can give them out in different patterns, but the cards are the cards, okay? So in your textbook, they talk about heritable features varying among organisms, such as flower color in this case. That's what they call a character. Okay, and this gets a little confusing because a character is the same thing as a gene. So when we use, I personally use the word gene almost all the time, but character and gene mean the same thing. But as you know, there are variants, right, to these genes. And I generally, basically almost always call them alleles, okay, but your book likes to refer to them as traits. So for example, so here you can see this purple flower, but it does come in another trait or another allele, which would be white. Okay, so the gene here that we're talking about is the flower color gene, and the traits or the alleles would be the purple or the white variant. Now, Mendel got extremely lucky. Well, maybe it wasn't even luck. Maybe he really thought this through. But he chose 7 distinct characteristics to study in his pea plants. Okay, if he didn't study these 7 traits right here, we might not have known as much as we do today about genetics. And the reason that this is so important is because these 7 traits, they're an either or. We're going to get into some really complex stuff with genetics later. Well, in Bio 1 Honors, we don't go too deep, I guess, but we're going to get into some complex patterns of inheritance. This, you're going to find out, is not very complex. It's an either or thing, right? Like, if I asked you guys, hey, what are the odds that you could do something like this? Like, take your thumb and can you bend it backwards like that? Okay, that's one gene that allows you to do that. Okay, some people can only go like that and they can't bend that thumb backwards. So it's either yes, you can, or no, you can't, right? For a gene like that. Well, that's kind of what these pea plant genes are. He didn't care how purple, he didn't care how tall, things like that. He just cared, oh, okay, it's either this or this. Like, for example, imagine if I was asking you about eye color. Think of all the different shades of eye color out there. We can't study that and think it's going to be simple. No, that's going to be very complex. So Mendel picked these 7 very simple traits to study. And as you can see, we have things like, well, flower color, flower position. Okay, axial is within the plant and terminals at the end. That's actually a gene. The seed color. Okay, so you have yellow or green. The seed shape is going to be either round or wrinkled. These kind of are missing some water, so you can see how it kind of prunes up on itself. The pod shape is either going to be inflated, or you can actually see the peas in there, constricted, we call that. Now the pod color, okay, is green or yellow. And then the stem length of the plant. So the stem is either going to be a nice tall stem, or it's going to be a short, or what we call a dwarf. Okay, so short. And when we get into it later, or if you need help understanding what's dominant, what's recessive, this is all dominant on this side, and this is all recessive over here. And it can get confusing. For example, look at seed versus pod color. They're flipped. Okay, so it's very important to know which is dominant, which is recessive, but we'll get into that later. Okay, let's take a look at a flower here. Okay, so what Mendel did was he took a flower, and as you can see here, there's of course the beautiful petals, but he took the male parts off. Okay, these are called the stamen right here. Okay, and they're the things that release the sperm. So a lot of us get, hay fever in the spring and things like that, and we're inhaling plant sperm or pollen. Okay, that's where these come from. So the male seeds come from right here. And then this carpel, okay, this is the female part. And down here, there's eggs inside of it. Okay, so you have to know a little bit about plant reproduction to understand how Mendel's experiment worked. So theoretically, this flower right here, part of a pea plant, could self-pollinate, right? Some of the sperm or the pollen that's right here could actually enter into the carpel and fertilize the eggs. Okay, now that's a weird thing because it's not asexual theoretically, even though it's one parent, because you're still mixing up 2 different sex cells. But very, very odd. So Mendel knew about this, right? And what Mendel did was he said, well, if I'm going to control mating in a controlled experiment, I'm going to have to make sure I know who mates with who. Okay, and how he did that is he took a scissors and he cut off the male parts. Okay, sounds wonderful. And by removing the male parts, okay, now you know that a plant isn't going to have those pollen seeds, right, those sperm cells, being able to self-pollinate. Then he took a paintbrush and he could paint whatever plant he wanted. In this case, maybe he cut off the anthers here, the stamen from the purple flower, and he took all the pollen and he started dusting it, okay, with a paintbrush. And by removing, okay, the male parts, he knows that if this flower does produce seeds, and eventually if this plant produces seeds, he knows that it's going to be the seeds from a different plant. Okay, and as you can see in this case, he went and go ahead, and this is called a hybrid cross, you're going to see in a second. He paints the paintbrush with the seeds from the purple flower, and he brings it over and fertilizes this white flower here. Okay, so what is so important, right? Like if I could have a bright flashing light or something right now during a video, I would say it's this right here. Mendel chose to work with true breeders. Okay, that is huge. So what that means is if you allow self-fertilization, the plant will produce identical to itself over and over and over. A true breeder. Like, for example, you've probably heard of purebred, if we're talking about like a golden retriever or something like that. So a purebred dog means that, oh, though that is 100% that, breed of dog. Well, true breeding means for whatever gene you're talking about, you are guaranteed that the only thing you're going to produce is that trait. Okay? So, for example, don't forget that this purple pea plant over here had parents at some point. 2 different seeds came together. And what that means is that this pea plant got a purple seed from one parent and a purple seed from the other parent. And that's it. So it's basically like purple, we could call that, okay? Whereas this white plant would have gotten a white seed from one parent and a white seed from another parent. So he's white. All right? So true breeders are very important to understand that Mendel always started with true breeders for his experiments. Okay, and notice I said started. So what do you call this then? If we're going to mate true breeders together, the offspring are called hybrids. Okay, so when you do cross fertilize, so you don't want the same flower to go ahead and pollinate itself, right? So when you do cross 2 different flowers, that's called a hybridization. You're making hybrids, or just a genetic cross, right? And true breeding parents, okay, in this case, are called the P generation. All right. And their hybrid offspring are called the F1, okay, which is Latin for the son, filial. So what's happening here is these 2 parent plants are going to go ahead and create the F1 generation. Now it's hard to think of because here we see flowers and now I'm seeing peas. Don't forget these peas, that's like you, right? That's like a zygote or something. You're going to put this in the ground and boom, it's going to grow into a plant. So don't forget these seeds are actually offspring, okay? And then we're going to go ahead and say, all right, here we go. We have our F1 plants. We'll take a look at this cross in a second. And then if you mated those together, you would get your F2 generation, right? And that would be like grandkids of these parents up here. So let's think of the 2 people that made you. They are your P generation, and you are their F1 generation. And one day when you mate, you will create kids, perhaps, and that would be your F1 or your parents' F2. All right. So let's have a look here at Mendel's law of segregation. Okay, and this is pretty impressive that he figured this out. All right, now this is very impressive that Mendel was able to figure this out because he didn't even know what chromosomes were yet. Okay, we're going to get into that later. This guy named Thomas Hunt Morgan working with fruit flies is really the guy who figured out the chromosomal basis of where our DNA is and how we accept our inheritance patterns. Okay, but Mendel actually figured out segregation. Now, if you don't know what that word means, it really just means separation, right? Segregation has some negative connotations in our country, obviously, because of bad decisions made in the past, right, with segregating of people. But segregation in this case just means separation, okay? So separation of alleles, Mendel actually figured out anaphase of meiosis. He didn't even know what a chromosome was, right? He didn't know about the nucleus and the DNA inside of there and things like that. He just said that, okay, basically he figured out that we are diploids, okay, without using that word. He said, all right, everybody has 2 parents, and you get a copy of your genes from each parent, but you have to separate those genes, right, which we did last chapter. We have to only give 1 copy of the gene. We can't give 2. What allele will we give? Well, that depends. It didn't matter for him in the F1 or in the P generation here because they were true breeders. So, for example, is this parent going to give a purple or a purple? Well, who cares? It's a purple. And is this parent going to give a white or a white? Well, who cares? It's a white. But what he figured out was this, and we're going to solve this together in a second. He figured out that, all right, if I go ahead and I cross 2 true breeders in a hybridization genetics cross, meaning 2 different variant here, these true breeders, and we're looking at flower color. If I mated these parents together, I'm going to get an F1, and this is exactly what he got, that is purple. Now, right there, that should have blown Mendel's mind. He should have said, wait a minute, what about the blending hypothesis, right? I thought we were supposed to be like a blend of both of our parents. Okay, wouldn't I be like a purplish white or something like that in between. And people have questioned this in the past, right? A lot of times, for example, I'm much taller. My dad was pretty short, and my mom's pretty short, and I'm 6 foot, right? So it doesn't always work out that way. Now, some people would say, oh, there had to be something going on back in the day, or maybe there was, you know, a demon inside punishing the family or something. They're crazy thoughts back then. Now, thank goodness, we understand genetics. We understand the way that gene expression works, which we'll get into. So Mendel actually got all purple here in the F1, okay? Now, that wasn't surprising eventually to him, but as he got all of these F1s, he said to himself, well, where in the world is the white? I know I made it a purple and a white, and it's not like he did it once. He redid this experiment over and over and over again, and he realized that, well, white must be in there somewhere, okay, but I just can't see it. And this is how he started thinking about that deck of cards. He's saying, well, I know it got dealt white, but I don't know where it is right now. So what Mendel did, which is pretty genius, is he allowed these hybrids to go ahead and mate. So remember, in his F1, he got a whole bunch of plants here, okay? And now he said, all right, let's mate these plants together. So when he mated the entirely 100% purple F1, or F1 times F1, he got a very classic ratio, which is called a 3-to-1 ratio. 3 purple to every 1 white, okay? Or if you want to think of it as around 75% purple and around 25% white, right, is what Mendel was able to get there. And this is why that video, hopefully you watched it yesterday, was talking about Mendel's data is a little too much, because in nature, for you to get exactly, or Mendel was very, very close to getting 75% purple here and 25% white, which can happen. But I mean, that would be like if I gave you a quarter and I said, hey, everybody, flip this quarter 100 times. And if you all came back tomorrow with your data and all of you said, oh, you know, I got 48 heads and 52 tails. And you're, oh, wow, I got 49 and 51. And I got, you know, 53 and 47. I got 50-50. And nobody was beyond, you know, like 47 or 46. That's pretty unheard of. Yet that's what Mendel's data was. Now, come on. The guy's a wonderful man. Would he have made it up? I don't think so. So let's actually use some symbols here. Now, we're not really ready for a full-on genetics problem using all the vocabulary, but let's go ahead and use the symbols and figure out how I would set this up using something that we call a Punnett square. Okay? So if you printed this paper, you could do it right over here. Okay? But I'm going to go ahead and do it on a piece of paper, and you could do it wherever you want to. Okay? Maybe you, if you're doing it separately on a piece of paper, maybe you want to write like 9A, page 2, or something like that on it. All right, let's give it a try. Okay, so here we go. We know that for Mendel's cross that we've been talking about, it's the parents are purple and white. Okay, now I do apologize. My autofocus on my webcam is a little goofy here. But the first thing that you should really do whenever you're solving a genetics problem, and this is kind of introductory, we're not going too deep yet, is you have to know what's dominant and what's recessive. So to start off, you can either go back and look at that sheet, or a lot of times you can just bank on the fact that pigment is dominant over non-pigment. But it's better to know for sure. So to start off, we have to know that purple is dominant to white. Okay, and now that we know purple is dominant to white, the next step we should really do is think of symbols. Now, you can really get creative here and use any symbols you want, but to make it easier on your biology teacher or in college and things like that, actually making it easier on yourself as well, what we generally do, okay, as geneticists is, if we're not talking about a specific chromosome or in nature what's wild and not, what you do is you just take the dominant allele, okay, so the purple trait is dominant, and you use that letter and make it capital, okay? And then what you could do is you go ahead and make white not a lowercase w. A lot of people think that, but actually a lowercase p. So uppercase and lowercase. And if you want to know a cool story, go ahead and look up why uppercase is called uppercase, right? And lowercase is called lowercase. You might be surprised when you figure that out. So this means purple now, okay, this symbol, and this little p means white. And notice I know cursive, right, so I can write like that. But if I didn't know that, I would make my little p look a little bit different, because we're going to be sticking a lot of p's together. And if you just made one look a little bit bigger than the other, they might get confusing. So it's good to make one look a little different. So now that we come back over here, I'm about to show you something that's so important. And that's that Mendel started with true breeders. So remember, this purple plant is a true breeder for purple. And he had or she, two parents, right? So in this case, if you have two parents and you're a true breeder, meaning you know you're going to give purple to your kid, I would say that we should use big P, right? And if this plant over here is white, and you know you're going to give white to your kid, well, that would be little p, right? So that's actually our cross now. These are our alleles here, our purple, purple, and white, white. Now what we're looking to get here is our F1 generation. So this is the cross that we set up. Now what you really want to think about is, what could this plant right here, let's just give a sex to these plants. Let's pretend we're using the male cells from this plant. That's a symbol for a male, and then the female would be like this. So if we're given the male seeds, what could this guy put in his sperm? Okay, now don't forget, you make 4 genetically dissimilar haploid gametes, but in this case, because this is true breeder, the gene or the trait here doesn't matter. It's going to be big P. Okay, so no matter what, let's just look at this and say, well, he could put a big P in one of his sperm cells, or a big P in one of his sperm cells, right? And what can this girl over here put in her egg? Well, she could put a little p in an egg, or she could put a little p in an egg. And again, remember, girls do their meiotic events a little different, but that's okay. So how can we decide? Well, theoretically, you probably already figured out that the F1 generation, okay, is going to have a big P from dad in this case and a little p from a mom or a maternal cell. You're going to be 100% that. Big P, little p. Okay? Now, that means purple. Okay, right? Because we said big P is dominant over little p. There's a purple flower. And that's what Mendel was talking about. Okay, Mendel said, I know white's in there, but I don't see it. This is actually how, spoiler alert, he figured out dominance. Okay, but how we can solve these problems, now if you're in a different class, right, like for example my my Bio 2 honors kids or my AP Bio kids, I usually don't like them even using Punnett squares once we get good at this stuff, because the mathematics is much easier than doing Punnett squares. For example, we're looking at one gene right now, we'll be able to do a little tiny Punnett square. But for certain crosses, you would have to have like 256 squares or something like that. That's a lot, right? So the math is going to be important, and we'll get into a little bit of the math soon, okay? But here we go. To set up a Punnett square, this is by Reginald Punnett. Figured out this neat system, and basically all you're showing here is meiosis. So in other words, let's go back up to the parent for a second. This guy right here is either going to give a big P or a big P to his kid. Sure, let's go ahead and put dad up here. Big P, big P. This lady over here, she can only give a little p or a little p to her kid. Oh, and now what you do is you just fill in the square. What if, by the way, what this is that I'm about to do is called random fertilization, right? Remember, for a human, it was that, you know, 1 in 8 million shot of that sperm and a 1 in 8 million shot of that egg, and it'd be a 1 in 64 trillion chance of setting up your chromosomes the same way. We're just talking about this random sperm meeting this random egg, and if that happened, let's keep our dominant first. Big P, little p, big P, little p, big P, little p, and finally, big P, little p. So we get in our F1 100% big P, little p, or purple, thus negating the blended hypothesis. Now I'm going to tell you something else here that'll help you a little bit. This does not mean that we just got 4 kids. We did not just get 4 kids from these parents here, okay? What this means is that if this parent really did mate with this parent, these are the possible offspring that they could get. So it doesn't matter if they had 1 seed or if they had 5,000 seeds. Theoretically, they should all be big P, little p. They'll turn into purple flowered plants. Okay, these are not kids in here. That's very confusing. These are the events, that the random fertilization results basically that could happen. Okay, so if this was the sperm that was used and this was the egg, there you go. Okay, so this is your results of meiosis and random fertilization. Okay, so now Gregor Mendel says, okay, well, I just disproved the blended hypothesis, it would seem, where did the white go? So he decided to go ahead and move on to his F2 generation. He said, well, okay, I have these plants right here. Let's go ahead and let them mate. So remember, maybe he got, instead of 4, these are not kids, these are possible kids. Maybe he got, I don't know, 73 plants. Or let's just keep it easy. We'll say he got 100 plants. Okay? So if he got 100 plants, he's going to take any of those 100 plants and he's going to mix them together. He's going to take the pollen, mix it with the eggs, and he's going to let these F1 plants reproduce among themselves. Okay? So he took an F1 and he multiplied to an F1. Or you could even say he let one of these self-pollinate. Okay? He let it cross to itself. And what's going to happen here is that's going to get him his F2. Okay, so let's set up our F2 by making sure we understand the cross. It's going to be an F1 plant, and we're going to cross that to an F1 plant. Big P, little p times big P, little p, right? That's our F1. We're going to cross them together. And now let's take a look what we can get. Now, I personally would rather you thinking about meiosis. What could this plant put in its sperm? Well, we could put a big P in a sperm, or we could put the other allele, okay, or trait, the little p, the white allele. So even though this plant right here is purple, and this plant right here is purple, theoretically they could give white. Let's check it out in a Punnett square. So I'm going to take one parent and put them on top, and I'll take the other parent and put them on this side. And that means we get big P, big P. Big P, little p, big P, little p. Ah, now this one's little p, right? And if you look at that Punnett square right there, you can see that what we got here was a purple, a purple, a purple, and a white. So theoretically, these are not kids, don't forget. These are the possible kids. So we get about 3 purple to every 1 white. Theoretically, that's what we should get. So I told you, let's imagine again that these two mated, and let's just say they had 100 kids. So if they really did mate these plants, or we mated them, and you actually got 100 kids, what you should expect, based on this Punnett square right here, is you would say, well, Mr. Boylan, I'll bet you we're going to get about 75%, or 75 plants in this case, same thing, purple, and I'll bet you we're going to get about 25%, or in this case 25 plants that are white. Right? And that's pretty much what Mendel got. Now we'll talk about what would happen if you got like 70 and 30, or maybe 65 and 35. There's some cool statistical tests that you could do when you get a little older, and some of you might know about them now if you've already taken stat. Okay, there's this really cool test called the chi-square test that you can see if the data actually is a good fit. But we'll talk about that later. But what's happening right here looks pretty good. Okay, so there's our Punnett square, and that's how Mendel was able to find out, well, the purple and the white were mixed. Okay, I got hybrids. They're all purple. I really believe that white is still in there somewhere, because you're not changing these genes, okay, which he didn't call them genes, but he called them merkmals. But he goes and mates the F1 to themselves, and then whammo, there it is. He got white again. So Mendel actually disproved the blended hypothesis, and now he had to really sit back and think, what does this mean? Okay, so let's continue. Okay, so hopefully you were able to stick with me for that problem. Let's go ahead and keep moving on here. So for these results and others, Mendel developed 4 hypotheses. Okay, and we're going to use the newer words that we use today. All right, so there are alternative versions of genes. Mendel, of course, knew that right away. So, for example, flower color, there was purple and white. Okay, alternate versions, and we call these things alleles. So that's very important for you to be able to use that word allele. Now, here's 2 new vocab words that hopefully you looked at yesterday. For each character, okay, or gene, an organism inherits 2 copies, one from each parent. You know that already. It's like a diploid from a sexual reproducing organism. So an organism that has 2 identical alleles is, we just said, true breeding. Oh, but in this case, it's not true breeding. We just call it homozygous. Okay? The word true breeding simply was used by Mendel because, oh, we already know what you're going to give your kid. But that's just like saying homozygous, right? Homo means same. Okay? And zygosity is talking about your genes here, or your traits. So, homo same gene. Oh, okay. So that means that the purple flower, for example, was big P, was a homozygote, and the white flower was a little p. He was also homozygous. Okay. So an organism that has two different alleles for the gene is said to be heterozygous. Hetero meaning different. Okay. So if you're displaying heterozygosity, let's take a look up here again. Oh, that would be these people right here. Remember in our problem, we got big P, little p. That plant technically would be a heterozygote. Okay, and that just means, okay, you have different copies, okay, of whatever that gene is, different alleles. Now this one seems, very obvious to us today, but back in Mendel's time, this was pretty important, right? If you have two alleles, the one that determines the appearance is called the dominant allele, okay, and the other one that doesn't really have an effect is recessive. For example, now that might be easy to you, but you have to wrap your head around this. A big P purple plant looks exactly the same way as a big P, little p purple plant. Okay, so that would just be dominance, right? Purple is dominant. We're going to break that rule eventually and get into some complex inheritance patterns, but for right now, dominant just means the allele that's expressed all the time. So how do you ever look recessive then? Well, to look recessive, you would have to have two copies of the recessive allele. The only way to be white is to not have purple, right? And then finally, Mendel figured out, this is so cool, we spent a long time doing last chapter, he figured out segregation. He figured out anaphase and meiosis. He said, sure, okay, so we pass on some sort of sex cells to our offspring, but you know what you have to do before you can do that? You have to make those cells correctly. You have to segregate your genes, your alleles, right? So let's just pretend that you are big P, little p. Well, you have to only give one of those to your kid. Are you going to give the big P or are you going to give the little p? Who knows, right? That's how you line them up together. So that's pretty crazy. But segregation, Mendel figured out anaphase of meiosis, which is crazy, right? So some more vocabulary words that are super, super important. Because an organism's appearance does not always reveal its genetic composition, geneticists distinguish between an organism's observable traits, which we call the phenotype, okay, and its genetic makeup, which we call the genotype. This is so simple. You're sitting there right now, right? Think about what color your eyes are, all right? Let's just say they're like mine. They are blue, okay, gorgeous blue, or maybe they're pretty brown or horrible green, or I'm just kidding. So that's your phenotype. Okay, your phenotype is basically the expressed or the physical, expression of your gene. That's your phenotype. The genotype are the actual nucleotide sequences of your DNA that we gave the symbols to. So the genotype, for example, you might be big P, little p. That's a genotype, right? Big P, that's a genotype. So phenotype is what you see, and genotype is basically the letters or the genes that make that up. So, for example, think about your hair color right now, right? Think about how tall you are. Think about the color of your skin, right? They are all phenotypes. Think about big E, little e. Think about big T. Well, those are genotypes, right? You wouldn't even know what I was talking about necessarily. So now you know for the F2 plants, the ratio we can get into as well. So remember I said we got about 75% purple and 25% white? Well, that would be a 3-to-1 phenotypic ratio because purple to white. Whoops, let's click the word. It told me access denied. I don't know what's going on here. All right, give me 1 second. Okay, sorry about that. I don't know what happened there. So what's happening here then with genotype? Let's think of that. If you had a 3-to-1 phenotypic ratio, right, in the F2 of that problem that we just solved together, 3 purple to 1 white, the genotypic ratio, right, if you're looking back, that would have been a 1 to 2 to 1 ratio, and we'll practice these together. But I will show you what I'm talking about here real quick. So if you remember, we ended up with this many purple and this many white, 75, 25. Oh, in other words, let's just think of what these plants look like. 1, 2, 3 were purple and 1 was white. Now we can actually say that is our phenotypic ratio. Okay. Our genotypic ratio then, let's take a look for that one. Right? Genotypic ratio. Okay, well, let's see. How many big P's could we possibly get from these parents here in the F2? 1. 1 big P. How many big P, little p's could we get? Well, we can get this one or this one. That's 2. And how many little p's could we get? Well, just 1. Oh, okay. So my phenotypic ratio is a 3:1, and my genotypic ratio is a 1:2:1, just what the letters are. Okay, that's all. Pretty easy stuff, but those vocab words are very, very, very important. Okay, so moving right ahead here. Mendel found that each of the 7 characters that he studied, okay, exhibited the same inheritance pattern. Okay, in other words, the dominant and the recessive were mated. That's a hybridization. The recessive always disappeared in the F1, and then it showed up again in the F2. And that's what led him to understand segregation, basically. And this applies to all sexually reproducing organisms, including us. So this should be a review from last chapter as well. You know what homologous chromosomes are. They bear the alleles for each character, right? So if you look here, let's just say that these are your homologous chromosomes, okay? And one of them came from dad and one of them came from mom. So these would never connect here at the centromere. That's not what these would do because they're homologous, right? Sister chromatids would connect at centromeres, not homologous pairs. They might cross over, but that's getting way complex. So let's just take a look at a chromosome from mom and the exact same chromosome from dad. And if you notice here, oh, look at that. It's homozygous for this gene right here, the P gene. Okay, let's call it. Well, this person, let's pretend this is you. You are homozygous for P because mom gave you a dominant P, dad gave you a dominant P allele. Okay, so you're big P. And that's the locus for the P gene. Now let's look at a different locus here. This is the A gene. Hey, you're also homozygous, or basically you're a true breeder for these genes, right? Because you're little a. Okay, and now out of these loci, let's look at one that's different. Here's our third locus. Oh, look at this one though. Mom gave you a sequence of nucleotides in your DNA in here that would tell you, hey, you're big B. Whereas dad's, sequence of DNA said, no, little b, you're recessive here. Or think about the flower. It's almost like you're purple, white. Okay, so you'd be purple. Okay, but in this case, it's big B, little b, so you're a heterozygote for that. Okay, you're not a true breeder. Certainly, when you go to have kids someday, are you going to give the big B that your mom gave you, or are you going to give the little b that your dad gave you? Who knows? You're a heterozygote, so it's like a coin flip. Okay, so now we're going to move ahead to something that's a little bit tricky for kids to wrap their head around, but as long as you know how to FOIL, okay, you should be just fine. But let's take a look at the law of independent assortment. Okay, and all this is, we're going to do the same thing that we just did, only we're going to look at 2 characters at the same time, or two genes basically at the same time. Okay, so this one is a little bit tricky, all right? And all Mendel was really trying to figure out here is, are genes always basically stuck together? Are they always inherited together? Or are these deck of cards thing, right? Could you separate them and could you deal some cards out, okay, and keep it in a hand? But could another hand, if you want to think of it that way, have a different set of cards? It's very confusing. So he said, for example, let's look at a pea plant that is round and yellow now. Okay, and we're going to cross that to a pea plant. Instead of the round seeds, you remember the opposite of round? It was wrinkled. And instead of yellow seeds, we're going to do green. So we're going to cross a round yellow, completely dominant pea plant to a wrinkled green, completely recessive pea plant. Okay? Now what Mendel's saying is that, all right, I want to find out, do genes sort themselves independently? Right? Are they their own individual units, or are they stuck together? All right, so let's take a look here. So how he figured this out was to do something called a dihybrid cross. It's a very classic genetics cross that all kids that go through Bio 1 had to do a dihybrid cross. And what we're doing here, instead of just saying, hey, remember we just did big P times little p, and we got all big, little? Well, we're just adding another gene in there. So we're doing big R, big Y times little r, little y, little y. And that's going to give you big, little. Okay. And if you notice, this is round yellow times wrinkled green. This F1 would look round yellow. And that makes sense, right? You disappear. The recessive trait disappears. Okay. And then if we mate the F1 to itself, maybe we'll get a certain type of ratio. So here's what Mendel thought would happen. he thought, yeah, I know what's going to happen. You're going to make these F1s, and then you're going to go ahead and cross them, and you're going to get a 3-to-1 ratio, yellow and round 75% of the time because the genes are stuck together, and then you're going to get 25% of the time you're going to get wrinkled green. This is not correct, okay? What we found out was that genes are their own units, right? Mendel called this independent assortment of genes. Okay, remember how they're all on different chromosomes, and in metaphase 1 of meiosis we line them all up in crazy different ways? Well, there you go. Mendel actually figured that out, and we're going to get this really classic 9:3:3:1 ratio by doing this cross together. Okay, now I just want to mention this now, and I'll mention it again in a little bit. This rule is definitely broken all the time. Okay, Mendel wouldn't have figured this out. And that's a thing that you'll get into with Bio 2 Honors or AP Bio someday, or maybe in college. You know, linked genes, well, you'll understand what those are. That just means if genes are on the same chromosome, then they actually are stuck together. But Mendel got lucky because his genes were on different chromosomes or far enough away that it didn't matter. So we're going to go ahead and show how this parent and this parent got mated to form this F1, and why when we cross the F1 we don't get something like this, but we get something completely different, because in this case the genes are not connected to each other. Okay, so again, we're going to use this page here. If you have it printed, you could do it right here, and if you're using pieces of paper, I'm actually going to make 2 gigantic Punnett squares. Okay, here we go. Okay, so here we go with Mendel's classic dihybrid cross. Now again, this is a 2-gene cross. We're not doing a single gene this time. We're doing 2 genes at the same time, and that will change up meiosis now, right? We're not just looking at one possible random fertilization event between one gene. We're going to look at it happening twice here. Okay, now like I said before, to start any genetics problem we really should know what's dominant and what's recessive. Alright, so in this case you could probably tell just because the symbols are already given that round is dominant over wrinkled. Okay, and then you also know that in this case we're talking about the seed. Yellow is dominant over green. So obviously round is going to be a big R, okay, and then a little r would mean wrinkled, and yellow is going to be a big Y, whereas green is going to be a small y. Okay, so let's just go ahead and set this up now. Something that'll save you some heartache is remembering you only ever put one gene above a block or to the left of a block in a small Punnett square, but when you're doing a 2-gene cross, you're actually going to have 2 alleles above or to the left of the squares. Okay, let me show you what I mean here. So we're actually going to make a 16 square Punnett square here. Okay, and I hope this shows up okay for you guys. Alright, let me go up here. Alright, close enough. So there's our 16 square Punnett square. Now, it doesn't matter what parent you start with, and for what we're doing right here is we're getting our F1. Okay, so this is going to be our F1, our inside of these. These are our possible kids, right? So let's start with this parent. And what I'm going to remind you to do here is you can FOIL it. Okay, so remember how to FOIL. FOIL means, let's take there's 2 genes here, right? Is a seed round or wrinkled, and is it yellow or green? Okay, the allele for this one happens to be round and yellow. They're true breeders. Mendel always started with true breeders. Okay, so round, yellow, FOIL. The first R with the first Y. Let's put this guy up here. There's my F for FOIL. First R, first Y. Now let's go to O. My outer R with my outer Y. My inner R with my inner Y. And now my last R with my last Y. So you're basically just distributing, right, your R's with your Y's. This R with both Y's and this R with both Y's, right? And now we're going to do the same thing for the other parent. So we took care of this parent. Now let's go ahead and take care of this parent. Now we have first R with first Y. Okay. And now the outer R with the outer Y. And then, right, you can see how these are all going to be the same. So to start off, you have to know how to FOIL. Now, this one was pretty easy to set up because these parents are true breeders, so we knew what was going to happen there. Now, when you're filling in these blocks, you go ahead and you keep your capital letter first, but you keep your genes separated is the easiest way to see what's going on here. So, for example, I'm going to bring down my big R, but now I have to bring over my little r. Now, where's my, okay, I do have a big Y. Let's bring that down. And now we bring that over. Big R, big Y, little y. And it doesn't matter what block you pick. Let's do this one. Bring down my big R, bring over my little r, bring down my big Y. Okay. So as you can see here, 100% of these blocks are going to be big R, big Y, little y. Okay, let me fill them in for you. 1 second. Okay, so now we're all set up here. And as you can see, every single block is the same. Now, does this mean that these two parents up here had 16 kids? No, absolutely not. What it means is that if these two parents, okay, reproduced, this guy could make these sperm, let's say, and this girl could make these eggs, let's say, and through random fertilization, they could come together to form these types of offspring. Now, there are 16 squares here, so it's 16 out of 16 are going to be round and yellow, but now they're both heterozygous, or what we can call dihybrid. Okay, so my F1, okay, is actually 100% big R, big Y, little y, or what we could say round yellow. All right. Now, Gregor Mendel at this point probably wouldn't have been too concerned. He would have said, oh, yeah, no, of course, this makes sense. All right, because I knew wrinkled and green would disappear in the F2. I'm getting good at this stuff, right? So what he's going to do is he's going to go ahead and go to the F2. How's he going to do that? You're Gregor Mendel, you're in the Abbey Garden, right? You're going to go ahead and take these plants, of which there might be 100, there might be 300 seeds, right? There might be 10 seeds, we don't know. But you're going to take the seeds of this cross, and inside of those seeds, what are the genes of interest? Well, they're going to be like this. We're going to mate them together, or let it self-pollinate. So let's mate two of these together to get our F2. All right. So now the factoring is going to be a little different here. So to get our F2, you know that we're going to go ahead and cross our F1 together. So we're going to do big, little times big, little, big, little. This is going to get us our F2. All right. And let's go ahead and set up our 16 square Punnett square, and basically all you're doing here is you're setting up your Punnett square so that you don't have to do the math behind it. And the math you're going to see as we go along is super easy. It's just what are the odds that you're going to give a big R, what are the odds that you're going to give a little r, and what are the odds that this person gave a big R or a little r. Like, it's just probabilities. It's like flipping coins, right? But we're going to stick to the tables for now, okay, to get good at them. So here we go. Now the foil is going to be a little different, right? Help me out here. What would this parent, let's put this parent on top right here. What would be above these blocks if we use the FOIL method? All right. Remember, when you're talking about FOIL, you're talking about the R's and the Y's. So the first R and the first Y, the outer R and the outer Y. So go ahead and pause the video and try and fill this in yourself and see how you would have done. Okay. All right, here we go. So I'm going to take my first R and put it with my first Y. Now I'm going to take my first R, my outer R and put it with my outer Y. Aha. Now I'm going to take my inner R and put it with my inner Y. Now I'm going to put my last R with my last Y. All right, so I distributed my R's basically. All right, and now I'm going to do the same thing for this parent. First R with first Y. Outer R with outer Y, and as you can see, they're the same parent, so of course it's going to end up being the same thing going down the side. All right. So there's that. Now we go ahead and we fill in this Punnett square. Okay, so again, try to do it on your own for a second, and what you should do is keep your capital first and keep the R's and Y's separated from each other. So go ahead and give it a try, and then I will fill it in. So let's pick one random block together to do together, so you know what I mean. Let's do this block. Okay, so I'm going to bring over my big R and, oh, it's a big R and a little r. So big R, little r. Now I have a little y and a big Y, little y. Oh, you've sunk my battleship. All right, so there's that one. So go ahead and fill in the rest of these, and let's see how you did. And magically we are back with it filled in. Okay, how did you do? Hopefully okay. So now what we did here was we really just took meiosis, right? What could this guy put in his sperm? What could this girl put in her eggs? And then we did the random fertilization here. And there are 16 squares. Does that mean we got 16 kids? No, no. These are just the chances, right? The possible offspring that you would get from this mating. So that's our F2. And Mendel thought he would get a 3-to-1 phenotypic ratio. He thought, yeah, I'm going to get about 75% of the plants are going to be round and yellow. And then 25% are going to be wrinkled green. Let's see what he actually got here. Okay, now here's a little hint. When I do phenotypic ratios for these, I like to go slow and make sure I write down everything that I could possibly get before I even start. Okay, now this is just my way, but it really helps out to really understand it. So for example, going down here, let's just say, hey, what could I have gotten? A round yellow times a round yellow. Well, obviously, one of the options I might get is round yellow. I'll bet you some of these plants are going to be round and yellow. Now, what else I like to do is I like to write down what that would mean looking at the letters, just to keep myself fresh, right? In other words, I know I'm going to need a big R to make it round, and because I'm doing phenotype, I don't really care about the other letter right now. And yellow, I know I need a big Y, and because I'm only looking at the phenotype, not the genotype, I don't even need to care about that. Because big R, no matter what, will be round, and big Y, no matter what, will be yellow. So I might get round and yellow. I also might get round and green. And I hope you know for that one, it would be round again, right? But now green, would be 2 little y's. I might also get what? I could get wrinkled yellow, right? And I hope you know that that would be little r, big Y something. Or maybe I'll even get some wrinkled green. And you know, wrinkled green would be little, right? There's no dominant at all. So here we go. Let's go through our Punnett square and find out, did we get any round and yellow? Now, Mendel was expecting a 3-to-1 ratio here. Let's go ahead and count how many round and yellow we got. We just need a big R and a big Y. So, 1, 2, 3, 4, 5, nope, 6, nope, 7, 8, nope, 9, nope. Okay, it looks like we got 9. I'm going to go ahead and recheck that. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yep. Okay, so we actually got 9 round and yellow. So a little over 50%, right? Because there's 16 squares. So 9 were round and yellow. Let's go ahead and check out round and green here. Okay, so round and green. Well, we know that that's going to be a big R, something, who cares, and now 2 little Y's. So let's check out round and green, and we're already done most of our things here. We don't have too many left, so is this round and green? Hey, it is. I'm going to go ahead and circle it in red. Round and green. No. Right? We actually ended up with 3 round and green. Okay. And what about, let's take a look for wrinkled yellow. So no capital R, but yes, we still want yellow here. Okay, and I'll use blue here. I'll just put a star in case you're colorblind. So I can see, wrinkled yellow, wrinkled yellow, wrinkled yellow. Nope. 3 wrinkled yellow. And we only have 1 left here. Hopefully it's going to be wrinkled green. Let's see. Wrinkled green. Ah, yeah. So I'll go ahead and let's make a squiggly line under that or something. All right. So technically, what we just did was we figured out our phenotypic ratio. Right? Phenotypic ratio is 9, 3, 3, 1. Okay. Now Mendel thought it was going to be a 3-to-1, round yellow to wrinkled green, but because we got this right here, round and green and wrinkled and yellow, Mendel said, oh wait, that shouldn't have happened. Oh my gosh. These genes are actual individual units. You can actually, they're not necessarily stuck together, right? You can either give a little r and a big Y, or a little r and a little y, or a big R and a... Wow. So Mendel actually figured out that genes assort themselves independently by setting up this dihybrid cross. Genes are not stuck together. Okay? Now, again, as you move on with biology, you're going to find out that, well, sometimes they are, of course. If they're right next to each other on the same chromosome, they're likely to be stuck together. But for right now, Mendel didn't know about chromosomes or any of that stuff. He said, look at that. Genes are their own individual unit. So let's pretend that I had you had, $100 in your hand right now, a $100 bill. And you had to place a bet. No matter what, I said to you, well, here's what happened. We mated these two plants together, okay? And what I want you to do is you have to make a bet. And it doesn't matter, the odds or anything like that as far as how much you win. If you're right, you double your money. And if you're wrong, I get your $100 bill. So if I mated these two, what bet would you make? What would the kid look like? Well, you're probably going to take that bet right there, round and yellow, right? So that would be 9 out of 16. So your odds are a little bit in your favor, right? And you probably wouldn't have picked one of these, right? Because the odds are very bad on this, okay? And another thing I often ask kids is, hey, what if I really did mate these two plants here? And I took all of their seeds and put them into a bag, okay? So let's pretend I have a bag right here. How many seeds are in the bag? And I'll have a lot of kids saying, 16, and no, we don't know how many, right? However many seeds they produced, I don't know, okay? But it's not necessarily going to be 16, okay? It could be any number, all right? So then when I say to the kids, all right, let's pretend that just by chance there is 16. How many of those seeds are going to be wrinkled green seeds? And a lot of kids will yell out, 1 out of 16 will be wrinkled green. And I say, maybe not. How do you know? This is just what could and should happen. But that doesn't mean that if you have 16 seeds, you theoretically could have a bunch of these in there or none of these. Right? It's all random chance through meiosis. And that's why we're going to talk about probabilities soon. Okay? So, regardless, getting back to the main point of this, we just did Mendel's classic dihybrid cross. We looked at not one gene, but two genes now. Okay? And we started off by saying what's dominant, what's recessive. We foiled our parents to put them on here. Now, it's much different because when we did our small Punnett squares, remember, oh, that's only one letter above and to the side. But when you're looking at two genes, okay, or 4 total possible alleles here, now you're going to have 2 letters, okay? And we foiled them out, and we found out that in the F1, yep, the recessive disappeared. And now when we move ahead to the F2, we find out that, okay, well, let's foil these. Wow, we get a really funky ratio here, 9, 3, 3, 1, or if you don't really understand what that means, 9/16, 3/16, 3/16, 1/16 chance of those offspring right there. Pretty cool. All right, I know this is going on a long time, so I'm going to go ahead and cover one last thing with you here today. Okay, so after we just solved that dihybrid cross example, you can read this. Now, this is a little tricky to wrap your head around, but what it's basically saying is if you actually just looked at the round or the yellow, you'd find out that you got about 12-4 ratios, which actually reduces to 3-1. So the round versus wrinkled was actually just a single gene that was kind of the same cross that we did today. Together we got a 3-1, but then the other was yellow, right, or green. That was also a 3-1. So when you combine all of that together, you get this classic 9-3-3-1 ratio. Okay, pretty cool stuff right there. All right. So, and that's how Mendel came up with the law of independent assortment, was by looking at two genes at the same time. All right. Last part for today. Here we go. Now, most kids pick right up on this, and if you're not one of them, that's okay. All right, we'll get you there. But what we're going to do now is we're going to find an unknown genotype. We're going to run something called a test cross. And as you can see, we have 2 little snake nuggets here. We have the burned variety, and then we have the frozen regular variety here. So what's happening is, and actually Pike 2 is doing well upstairs. He just ate again this morning. So assume that in mice, black fur is dominant to white fur. Suppose you have a white mouse. You can then confidently assume it's little b, right? If black is dominant, then this guy right here has to be little b. But what about him? you know that he could be big B or big B little b. How can we find out? Now, in today's world, you could probably get a genetic test done where they read your sequence of nucleotides and tell you exactly what your gene is or your alleles. But let's say that we don't have that option. So how can we figure out if he's homozygous, big B, or heterozygous, big B, little b, because they would look alike? Well, we could mate them, okay? And this is what scientists do. And again, it's ethical because they're mice, right? You can't do this with humans. Like, you over there, mate with you, and let's wait, 12 years and find out what the kid actually looks like. No. The mice, yeah, you put them together and you can mate them and take a look. And think about what's going to happen here, right? This is called a test cross. Whenever you made an unknown genotype, big B times little b, or big B times little b, you're trying to look at the kids and find out what was this parent. Okay, so think about what we're actually doing here. What are we expecting to see in the kids? Well, think, if he was big B, it doesn't matter. All the kids are going to be black, right? Because he has no choice. He has to put big B in his sperm. So she has to give little b in her eggs. So big B would only produce black mice. But what if he was big B, little b? Now half of his sperm would have the big B and half of them would have the little b. And if he gave the little b to half of his kids, theoretically, well, then she gives you could end up with white. So if you see any white offspring, you know that he was a heterozygote. All right, let's try it out here. Okay, so 1 second. Okay, so part of me is glad that we're not in school, because when I draw these things on the board, I get a lot of laughs. I'm talented at just about everything in this world except for drawing mice. I'm just not good at it. So if you don't understand what these gorgeous pictures are, this is the black mouse, right? And here's the white mouse. And as you can see, he's either a homozygote or he's a heterozygote for the coat color gene. Okay, and then for the coat color gene, this mouse is a homozygous recessive, right? Because the only way to be white is to be little b. So test cross, here we go. If we mated the black to the white, right? And we get, let's just say this. Let's say that we got, let's say 7 total offspring. Let's say that out of 7 total offspring, 3 were black and 4 of the pups were white. Well, if you did a test cross and you got these results, you could be 100% now that the black mouse was what? Heterozygous, right? Big B, or big B, little b. Now, that's a test cross, right? So if I had to solve the Punnett square, you would have said, yes, look, Mr. Boylan, here's how it happened, right? That would have been the black mouse. This would have been the white mouse, okay? And then when you cross them, you'd see that about half of the mouse were that, and about half of the mouse, or half of the mice were that. You get about a 50-50 ratio. And hey, look, we were pretty close to that, right? Should have got 3.5 if we were Gregor Mendel, right? But it's hard to get that. So point is, yeah, the test cross showed us that the black mouse was that. Now, it does get a little funky, right? What if we did this? What if we mated the black to the white, and we got, let's just say, they had 6 kids. Let's say 5 pups. They had 5 black in the F1. Oh no, what are we going to do here? Can you confidently say, because they had 5 black pups, that the dad had to be this? Or in the back of your head, are you saying to yourself, yeah, but he could have been this, and maybe just there's a chance that for every one of those kids, he just gave the big B. It's unlikely. He probably should have given a little b half of the time, but maybe, have you ever flipped a coin 5 times and gotten heads all 5 times? It happens. So, what do you think a scientist would do here if he only got black? Do it again, right? Do it again. Lucky guy, let's mate him again. Maybe find another white mouse right away and go ahead and mate him. And now in the second mating event, okay, so same cross, maybe he had 4 pups and they were all black. Whoa, now you're talking about 9 total mice that are all black from a black to a white cross. Now are you confident enough to say he was big B? I would be. But some scientists say, let's do it again. What is the magic number? When can you be happy enough to say, yes, I am sure that this guy was big B, and that is determined by your experiment, right? Your sampling size has to be big enough for you to be confident. And there's these things as you get older called confidence intervals and different statistical tests that you can do to actually answer that question. Okay, because you might be confident, or if you do it 1,000 times, you might be very confident, right? And we'll get into that stuff later. So, and it's really neat when you get into that stuff. For example, a field biologist has to be confident, maybe 95% of the time you're confident in your data. Okay, whereas maybe if you're like a pharmacist or something like that, creating drugs, right, or maybe the vaccine, for example, as you know, that's important right now. So maybe you have to be like 99.99% confident before you start injecting stuff into people's bodies, right? So we'll talk more about this as you get older and as you go through more biology classes. But point is, a test cross is when you have a known phenotype, black, okay, but an unknown genotype. Big B or big B little b, we don't know. So how can you find out? You test cross it. You cross it to a homozygous recessive, and then you look at the kids. If you get any white kids whatsoever, you know that he had to be a heterozygote. And if you don't get any white kids, well, he's probably then homozygous dominant. Okay, that's a test cross. Okay, Beaks, so I hope that was helpful with the test cross. Now here we go. Practicing what you've learned. We covered a whole lot of genetics today, and it's all rudimentary, simple stuff. Now, I'll be honest, if we were in school together, we might have just done the single gene crosses today, right, with the smaller Punnett squares and the one letter above and to the left. But we went ahead and we got into dihybrid or 2 gene crosses today, which has foiling and 2 letters. That's a lot, okay? And not only that, but you're at home. Here I am talking at my computer, and I'm used to being able to stand at the board in front of you, right, watching you all just marvel at the glory that is in front of the classroom. And that way you could stop me, you could ask questions. So I know this might be a little tough on some of you, but what I want you to do is I want you to try these 5 practice problems, okay? I'll be honest with you, we actually did this one together, but don't look at it that way. Try it on your own. The only difference is we're starting with a true breeding tall and a short, okay? So big T times little t, right? Okay, that's how you're going to set this up, but now it says get the phenotypic and genotypic ratios at the F2. Show all Punnett squares. All right, so go back and look at the first one that we did. Now here we're doing a heterozygous yellow-seeded pea plant to a green-seeded pea plant. Now you might say sometimes, well, how do we know? Is it heterozygous, homozygous? Unless I have typos in here, which as you know I don't make mistakes, that would be everything that you need is given to you. Okay, so you can go back and look what's dominant, what's recessive, but theoretically everything that you should need to know is in the question. Remember, start with what's dominant and then write out your parents and put the symbols down. This one says, imagine tall and short pea plants were observed growing in Mendel's garden. These plants were identically grown under strict environmental conditions. For which phenotype would you know the genotype? So this is really just getting you to explain, do you understand what those vocabulary words mean? Practice problem 4, oh boy. All right, this is a 2 gene cross, so this one will be foiling, okay? And it's not going to be that 9:3:1 ratio because I'm switching it up on you. Here we have a short heterozygous purple pea plant. So short heterozygous purple, what would that look like? So put the letters here, there should be 4 of them, right? And then a heterozygous tall and a heterozygous purple pea plant. Okay, so think about what that would be. Heterozygous tall, heterozygous purple, times 4 letters over here. And now you're going to have to do a 16 square Punnett square here. Okay? Now, it doesn't necessarily mean you have to go to the F2. That was just Mendel's cross. You're just doing one cross here. And finally, number 5. My goodness, my kids are upstairs so loud right now. I don't know if you could hear them. Or else maybe like a bear or a wolf got in. I don't know. Well, I'm not going up there to check. So practice problem 5, if you cross true breeding smooth seeded pea plants to wrinkled seeded pea plants, the F1 will be 100% smooth. Explain why the recessive phenotype disappears in the F1. Okay, so hopefully that might make sense to you and try your best on that. Okay, so tomorrow I'm going to go ahead and go over these problems with you, and I'll probably just give you maybe a couple more to work on or something like that because, again, genetics, you learn it slowly, methodically. You do your problems, and you don't want to move ahead too quickly until the ones that you're already doing make sense. Okay, so hopefully that was helpful. I'm going to get out of here and say goodbye. 1 second. Okay, Beaks, I hope that started you off okay. So the goal now is for you to try those 5 problems. Now, I'm not having you submit this because I have a feeling that there'll be a handful of you who are just not quite ready yet to do these simple problems, and eventually, don't worry, you'll all be fine. But what I'm asking you to do is try these 5, and then tomorrow I'm going to go ahead and solve these 5 with you on a piece of paper, and we'll do some more problems and things like that together. Maybe we'll start probabilities a bit. But I hope this video helped a little bit, and I hope that these 5 problems aren't too tough on you, and that's okay if they are because we'll go over them tomorrow. Okay? Have a good day. Take care.